Hi, my name is Shannon Moncier, and I am a PhD student in the biology department in Dr. Woo-Suk Chang's lab, and I will be presenting monitoring changes of microbial communities in the human gut microbiome with the use of fermented soybean products. So the gastrointestinal tract harbors various complex microorganisms such as bacteria, which can be crucial to our overall health and well-being, while some may also contribute to illnesses and diseases. The gut microbiome is unique to each person, and each person has their own specific microbiome. And the reasoning why is because there are numerous factors that can affect and shape the gut microbiome. Some of these factors include age, gender, and diet, and that's just to name a few. In our study, we are using a fermented soybean product to observe any effects or changes in the gut flora. Fermented soybean products are considered a functional food, which means that they are rich in nutritious ingredients, which can be found to be deficient in the human diet, and they also have a pro and prebiotic effect. The fermented soybean product that we are using in our study is called Cheonggookjeong, and it's a traditional Korean fermented soybean product that has reports of pharmacological activity when used. This includes decreasing inflammation and also blood pressure. But here in the United States, there is insufficient clinical research, which is why we want to provide functionality and safety of Cheonggookjeong pellets in the Western gut, as well as monitoring changes in the microbial communities and microbiome before, during, and after consumption. Figure 1 shows what our product looks like and what our participants took during our study. Figure 2 displays our 8-week-long study, with the stars representing our sample submission time points. I do want to point out that the first star is our time point T1, which is the fecal submission prior to consuming our fermented soybean product. 2, 3, and 4 are while the participants were consuming the fermented soybean product and submitting their fecal samples. And at the 4th time point is where the participants stopped taking the fermented soybean product and went 2 weeks without consuming to submit their 5th and final fecal submission at the 8-week mark. Figure 3 shows how we took those fecal samples and extracted the genomic DNA. Figure 4 displays our microbial composition at the phylum level in the gut microbiome. Here, the time points shown are T1, which is prior to consumption. T2 is the 2-week mark. T3 is the 4-week mark, and T5 is the 8-week mark. The time point T4, which is the 6-week mark, was very similar to T3, which is why it is not shown in this figure. Figure 5 shows the alpha diversity, which is the diversity within the samples. Figure 6 shows beta diversity, which is the diversity between the samples. So looking at our conclusion from our data, the most abundant phyla that we found are Firmicutes, Actinobacteria, and Bacteroidetes. These are the most predominant phyla and are also beneficial to the host. Our alpha and beta diversities indicated that our samples were becoming less diverse and more similar with the use and consumption of fermented soybean products. There have also been recent studies that suggest that the use of a soy-based diet can actually increase the population of good bacteria while also decreasing bad bacteria, which can explain why we are actually seeing a decrease in diversity, because there's actually an increase in the good bacteria and they're becoming more similar with each other. So that is the human gut microbiome with the fermented soybean product effects, and thank you for listening.